Welcome to Unit 4, Part 4 of Molecular Pharmaceutics, focusing on nanotechnology and targeted drug delivery systems, NTDS. This lecture is presented by Dr. Krishna Yadav. This video explores two gateways for drug delivery, the pulmonary route and the intranasal route. Both routes bypass first-pass hepatic metabolism and offer a rapid onset of action. The pulmonary route provides a vast surface area for absorption, while the intranasal route utilizes the nose-to-brain pathways. Intranasal drug delivery involves administering drugs through the nasal cavity for local or systemic effects. This route is preferred as an alternative to oral and parenteral administration due to its ability to achieve rapid drug absorption, bypass the blood-brain barrier, and avoid first-pass metabolism. The nasal cavity is organized into three functional regions: the nasal vestibule for large particle filtration, the respiratory region with a rich blood supply for drug absorption, and the olfactory region, which is key for nose-to-brain drug delivery. Nasal mucus is primarily water, mucin glycoproteins, and proteins. It provides barrier protection, moisturization, and immune defense. Mucociliary clearance is a function, moving cilia to transport mucus to the nasopharynx. Intranasal delivery offers advantages such as a large surface area, rapid absorption, avoidance of first-pass metabolism, non-invasiveness, and direct nose-to-brain transport. This allows for rapid onset, making it suitable for emergency drugs and alternatives to injection. Limitations of intranasal delivery include mucociliary clearance, which limits contact time, and the limited volume each nostril can hold. Enzymatic degradation, molecular weight limitations, pathological conditions, and patient variability also pose challenges. Nasal drug absorption is influenced by several factors. These include drug properties like molecular weight and lipophilicity, as well as formulation factors such as pH, osmolality, and viscosity. Physiological, anatomical, and delivery device factors are also critical. Absorption enhancers for nasal delivery include surfactants, bile salts, chelating agents, cyclodextrins, and mucoadhesive polymers. These agents, along with fatty acids and tight junction modulators, function to increase membrane fluidity or open tight junctions to improve absorption. Intranasal drug delivery systems include liquid formulations like nasal drops and nasal spray solutions. Drops are simpler but offer imprecise dosing, while sprays provide precise, uniform dose delivery via metered dose spray pumps. Semi-solid and solid formulations include nasal gels, which thicken aqueous formulations and increase contact time. In situ gelling systems transition from liquid to gel, offering prolonged retention. Nasal ointments and creams are used for local treatment. Solid powder formulations like nasal powders and nasal inserts provide improved stability and patient compliance. Powders are delivered via specialized devices, while inserts are solid dosage forms placed in the nasal cavity for sustained release and higher drug loading. Particulate and colloidal systems, such as microspheres, nanoparticles, liposomes, and nasal microemulsions, enhance nasal drug delivery. They improve residence time, protect drugs from degradation, and enhance permeation and bioavailability. Novel nasal delivery systems include mucoadhesive polymers like chitosan and carbopol, which adhere to the nasal mucosa to extend residence time and improve absorption. Drugs can also reach the brain through the olfactory pathway, trigeminal pathway, and systemic absorption, bypassing the blood-brain barrier. Nose-to-brain delivery systems have applications for treating diseases like Alzheimer's and Parkinson's, as well as epilepsy, brain tumors, and pain management, offering a direct route for central nervous system-acting drugs. Nasal vaccines target the mucosal immune site. Advantages include non-invasive, needle-free administration and the induction of both mucosal and systemic immunity. Examples are FluMist and Covaliv. Delivery systems include nanoparticles, liposomes, ISCOMs, and dry powder formulations. The preparation of nasal solutions involves 5 steps: preparing the vehicle, dissolving excipients, dissolving the drug in the buffered vehicle, adjusting the pH to 5.0 to 6.5, and finally sterile filtration and filling the solution into delivery devices. An example formulation is oxymetazoline nasal spray. Key ingredients include the active ingredient oxymetazoline HCl, the preservative benzalkonium chloride, and buffer components to adjust the pH to 5.5 to 6.5 for optimal stability and comfort. Preparation of nasal suspension sprays starts with drug micronization to a 1 to 10 micrometers particle size. This is followed by preparing the dispersing medium, incorporating the micronized drug and other components, and then homogenization and filling to ensure uniform drug distribution. Preparation of in situ gelling systems, like the poloxamer-based thermosensitive gel, uses the cold method. The sol-to-gel transition temperature, or T-sol gel, should be 32 to 37 degrees Celsius to ensure the formulation gels at body temperature, while maintaining easy administration at room temperature. Nasal microspheres and nanoparticles are prepared using various methods. Chitosan microspheres use spray drying. PLGA microspheres are formed by emulsion solvent evaporation. Nanoparticles use methods like ionic gelation for chitosan and nanoprecipitation for PLGA. Nasal liposomes are often prepared using the thin film hydration method. This multi-step process includes lipid film formation, solvent removal, hydration with buffer, size reduction via sonication or extrusion, and purification. Surface modification can also be performed. Physicochemical characterization is crucial for intranasal systems. Tests include measuring pH, viscosity, and osmolality to ensure comfort and stability. Drug content, sterility, preservative efficacy, and related substances are also monitored using various analytical methods. Performance parameters of nasal delivery devices are evaluated through tests like dose content uniformity, spray pattern, and plume geometry. These ensure the consistent and correct delivery of the drug, which is essential for efficacy. In vitro drug release and permeation studies are conducted to predict formulation performance. Release kinetics are tested using dialysis or paddle methods, while permeation studies use models with nasal mucosa to assess drug transport. Mucoadhesion testing evaluates how well the formulation sticks to the nasal mucosa. Methods include tensile strength, Wilhelmy plate, rotating cylinder, flow-through, and rheological synergism. In vivo gamma scintigraphy provides direct evidence of retention time. Nasal ciliotoxicity studies assess the potential harm to nasal cilia. The frog palate model measures the effect on ciliary beat frequency. Other methods use human or bovine nasal epithelial cell cultures, or the sheep model for in vivo assessment. Nasal irritation studies assess comfort and safety. In vivo methods include using animal models to observe changes in the nasal mucosa. In vitro tests use cell viability assays to quantify cell damage caused by the formulation. Pharmacokinetic studies, both in vivo and ex vivo, measure drug absorption. In vivo studies in animals or humans evaluate systemic and central nervous system uptake. Ex vivo studies use excised nasal mucosa to measure drug permeation. Stability testing monitors chemical, physical, and microbiological parameters according to ICH guidelines. Device performance, including spray characteristics and dose delivery at the end of life, is also assessed to ensure quality throughout the shelf life. This summary compares pulmonary and intranasal drug delivery. Pulmonary delivery targets the lungs with a vast surface area for high-dose systemic and local delivery, primarily using MDIs, DPIs, and nebulizers. Intranasal delivery focuses on the nasal cavity, offering rapid systemic delivery and nose-to-brain targeting using sprays and drops. These are the references used in this video, covering inhalation drug delivery, aerosol technology, and both general and advanced topics in intranasal drug delivery systems.